Welcome back to Chapter 5. We're going to do sections 4 and 5 in this video. So we'll start with sex chromosomes and sex determination, and then we'll finish up with dosage compensation of X-linked genes. So we finished up in the last video talking about the Y chromosome and talking about the functions that it plays in human males. And we're going to get a little more specific here and talk about how sex is determined in humans and the role of the Y chromosome. But first, let's start with a little bit of background here. Now, species that have distinct sexes are called sexually dimorphic, or they're said to have sexual dimorphism. And this is determined sometimes by chromosomes, such as humans that we already know. Females have two X chromosomes, males have an X and a Y. Some organisms, like turtles, the sex is determined by temperature, the temperature at which the eggs are incubated. So that's an interesting caveat beyond the scope of this course, but interesting nonetheless. Okay, so let's talk about sex determination in humans. What actually happens during embryonic development that leads to the gender of the offspring? Well, essentially, it's the presence of the Y chromosome that determines the gender of the fetus, and let me explain that. So if you have an X and a Y chromosome, as you know, that means you will be male. But on the Y chromosome, up here, there's a gene called the SRY gene. This SRY gene produces a protein called TDF. TDF stands for testis determining factor. And what happens is that this TDF protein, losing my cursor, goes to the primitive gonad. This is basically an organ that will develop into either ovaries or testes. This is the embryonic gonad, and essentially tells the embryonic gonad to develop into a testis, into the male gonad. The testis, in turn, will produce testosterone, which is a steroid sex hormone, the main male steroid sex hormone, and this is what's going to initiate the development of the male's sexual characteristics. Now, in contrast, in females, there are two X chromosomes. Now, because there are two X chromosomes, obviously there's no Y. If there's no Y, there's no SRY gene. No SRY gene, no TDF. No TDF means that there's no signal for this embryonic gonad to become a testis. In the absence of TDF, this is what urges or encourages or causes this embryonic gonad to develop into an ovary. The ovary does not produce testosterone, so in the absence of testosterone, the embryo itself will develop female sexual characteristics. So you could say that the female is kind of the default in the absence of the Y chromosome. Now, that's how it usually happens. Unfortunately, there are errors. Errors do happen sometimes. So sometimes we can have two X chromosomes that, for some reason, if there was an error, let's say in recombination, and the SRY gene ends up on an X chromosome, remember, the presence of that SRY gene is what starts the whole maleness thing in motion. And so you can have an XX male in this case. By the same token, if you had an XY embryo, and if for some reason that SRY gene is not there, in the absence of SRY, remember, we will not get a testis, we will not get testosterone, we will end up with a female. Now, there are a couple of specific disorders that we do see of sex determination in humans. The XX plus SRY giving the male phenotype, as I just mentioned, the XY without SRY giving the female phenotype, and also something called testicular feminization. Now, testicular feminization results from a mutation in the TFM gene. So what happens is the TFM gene produces the protein that acts as the testosterone receptor. Now, a receptor in this case, in biological terms, refers to sort of like an antenna that sticks out of the surface of a cell. So if you can imagine a radio needs an antenna to receive a signal. Well, cells need an antenna to receive signals, whether they're hormonal signals or any kind of signals that might be traveling through the bloodstream. Without that antenna, that signal may as well just not be there, kind of like with a radio. Without the antenna, those radio signals may as well not be there. So what happens in a normal male with a normal normal XY chromosome, you get that TDF being produced. But when that testosterone is produced from that testis, the TFM gene, which is actually located here on the X chromosome, which produces a testosterone receptor, has to be there on the testis in order for it to respond to testosterone and produce those male secondary sexual characteristics. If there is a mutation, however, in that gene, you won't get a testosterone receptor. So it basically is functionally as if there is no testosterone. If there's no testosterone, there's no signal to develop maleness, and you will end up with female secondary sexual characteristics. So you will end up with a phenotypically female fetus, which is actually XY. And a famous actress is actually an XY, and she actually was born with XY chromosomes, and she has testicular feminization. That actress is Jamie Lee Curtis. So go ahead and Google her. You can find out more. All right, now sex determination Drosophila. Now we talked about this a little bit on earlier videos, talking about Morgan's experiments. This is a little bit more, little bit different than what we have in humans. Now the Y chromosome does not determine sex. We know that from his experiments. But instead, the sex of the flies is determined by the ratio of X chromosomes to autosomes. So if you have an X chromosome to autosome ratio of 1, you are female. Less than 1 here, you are male, or 0.5, you are male. Anything down here less than 0.5, you are, sort of male, or you have maleness characteristics. Anything over 1, you are phenotypically male, and anything between 0.5 and 1 is really neither male nor female. So it's really the ratio as opposed to just the Y chromosome itself. Now, other animals have a similar system, similar to humans or mammals, where there's an XY system. In some animals, the ZW system works similarly, but kind of different. This takes place in birds, butterflies, some reptiles. In this case, it's a little opposite. The males are the homogametic ones, which means that they're the ones that have the same sex chromosomes, whereas the females are the heterogametic, which means they produce gametes with either one or the other sex chromosomes. And in this case, the female determines the gender of the offspring because she can give either a W or a Z, producing either the female or the female here or the male, whereas the male can only give a Z chromosome. There's also another example in other animals, such as honeybees, the haplo-diplo system, which essentially the males are always haploid, females are always diploid. So how does this work? Well, the females are diploid, and when they produce eggs, they produce haploid eggs. When those eggs are unfertilized and they develop into offspring, they develop as males. However, when you have a haploid male and a diploid female, and if she produces a haploid egg, which is fertilized by haploid sperm, you get a diploid embryo, which will develop into a female. Okay, so now we're going to move on to section 5, dosage compensation of X-linked genes. Now, what's interesting to think about is when you talk about a system in which an organism has 2 copies of each gene. So you think about that, right? We've got 22 pairs in humans. Each of our cells, or most of our cells, have 22 pairs of the autosomes. So we have 2 copies of each gene. And if we deviate from that number, we're going to cause abnormalities. So if we have too many chromosome number 21s, for example, we're going to have Down syndrome. So if that's the case, then how is it that males can have only 1 X chromosome, while females have 2 and everything is normal. There's got to be a way to sort of compensate for that dosage, to compensate for the fact that males don't have enough, or maybe females have too much of the X chromosome. So there's 3 possibilities, all of which we see in nature. Now, either the X chromosome in males actually functions twice as hard as those in females. We call that hyperactivation, and we actually see this in Drosophila. Another example, something we see in mammals, is that actually one X chromosome is inactivated in each female cell, and we're going to get back to that in a second. Or the X chromosomes in females can only work half as much as they do in the males, and we see this in the roundworm C. elegans. But let's talk about the X chromosome inactivation that we see in mammals. So this is something that is common to mammals. By inactivating one X chromosome in each cell, essentially we are compensating for the, you could say, excess of X chromosomes that females have compared to males. So now this inactivation is actually random in each cell. So about half of the cells will inactivate the maternally inherited X chromosome in females, and about the other half will inactivate the paternally inherited X chromosome. Now this inactivation choice is maintained in the cell descendants. What I mean by that is when those cells divide, the daughter cells will have the same X chromosome inactivated. However, all X chromosomes will be reactivated in the germ tissues, or the tissues that are going to develop into gametes. Now, so what happens is because the X chromosome is inactivated, we end up with genetic mosaics. Okay, so if we look over here, and a great example is coat color in cats. So coat color in cats is those genes are carried on the X chromosome. So let's say we have this zygote here, this embryo, female embryo of a cat, and we have an X chromosome here that has the allele for light fur, and this one over here has the allele for dark fur. Well, what happens is that if you look at the embryo, which is composed of a bunch of cells, in some of the cells we're going to have the inactive X chromosome is going to be the one that carries the light coat color allele, and in some cells we're going to have an activation of the other X chromosome, which carries the other coat color allele. So what's going to happen is that as that embryo develops, we're going to have sort of a spotting pattern, where you're going to have dark fur being developed by the cells where the active X chromosome contains that dark fur allele, and we're going to have patches of light where the cells contain that active X chromosome with the light color allele. And this is how we get sort of that patchwork pigment in these female cats. Now these are seen only in female cats, not in male cats, which is why calico cats are always female. Now this inactivated X chromosome, we can actually see it if we were to stain cells in females. We see something called a Barr body, which is a very small condensed dark staining structure. Now these Barr bodies, which is essentially the inactive X chromosome, what actually inactivates X chromosome is the addition of these chemical groups, these methyl groups, a carbon attached to 3 hydrogen atoms. When we attach those groups to DNA, it acts as a sort of like a repressor. It sort of turns the genes off like a switch, and it activates those genes. This concludes Chapter 5. Thank you for listening.